Uh,
Yes, we can hear you. Thank you so much.
Not yet. Oh, okay. Hello everyone. I'm Hao and Nansu Huang, and I'm very happy to present this literature titled "Effect of Source versus Household Contamination of Tube Well Water on Child Diarrhea in Rural Bangladesh," randomized control trial. And our presentation and slides were guided by Dr. Douglas Japes. And you can interrupt me at any time if you have a question. First, it's a brief introduction about the background of the study. The majority of rural Bangladeshis, they obtain drinking water from groundwater aquifers using the shallow tube well. We can see a picture on the right side of the slide. And depending on the depths of the shallow, there are shallow tube wells and the deep tube wells, and our study will focus on the shallow tube wells. We can see that the Bangladeshis, they use hand pump to lift the water from the deep soil. The groundwater is typically considered microbiologically safe due to natural pathogen removal and inactivation by percolation through the soil. But previous studies in Bangladesh in the 1970s, they failed to detect the reduction in rate of cholera and other diarrhea disease in tube well users. There may be two potential explanations. First, the water, tube well water, they remained sufficiently contaminated with pathogens to pose health risk. The second explanation is the water become contaminated during collection, handling, and storage in household. On the right side of the slide, we can see a container which was commonly used by Bangladeshis. They call it kashi, and we can see that it's a lidless aluminum vessel with a narrow mouth but a wide brim. Is there any comment on chat?
Yes.
Okay. We can see that lidless aluminum vessel with a narrow mouth but a wide brim. So it will leave the water vulnerable to contamination by contact with hands. So as a result, an estimate 16,600 to 27,700 children die each year in Bangladesh from diarrhea disease. And the investigators, they conduct a randomized control trial to evaluate the individual and the combined impact of safe storing and chlorinating tube well water on household water quality, and diarrhea among children who are under 2 years of age. They have two hypotheses. First, the children under 2 who drink treated and safely stored tube well water, they would have less diarrhea than those who drink untreated water. The second one is the chlorination lead to larger reduction in diarrhea compared to the simple safe storage. Let's come to the method part. They randomly select 87 village in the one of the district in Bangladesh. It's called Mamasan, and they have the pilot study about the dessert selection. That's because some district in Bangladesh, the soil have the iron concentration in the soil is higher than the other parts. And we know that we use one of the treatment is the chlorination. So we know that the iron may have a chemical reaction with the chlorine. So after the pilot study, they choose this district with lower concentration of the iron in the soil. The eligible criteria for the household was that they relied on a shallow tube well, which was shallower than 215 feet as their primary source of drinking water. They should also have no complaints of the iron processing their two wells. They will have a child between the age of 6 months and 18 months living in the household. And the household, they did not plan to move within the study period. At last, they select a random subset of 1,080 households from all the 2515 household that meets the eligibility criteria. The investigators, they use the randomized sequence using the random allocation function in the software of Stata. The study area was divided into 15 distinct regions. In each region, the household were listed in the order they were identified during the screening. And they used the block randomization with a block size of 3, corresponding to their 3 treatment arm. First is oh, okay. The first treatment arm is the chlorine pass plus safe storage. They use NADCC tablets and narrow mouth vessel with lid and tap. And I will show the picture of the container they use in the following slides. The second treatment arm is the safe storage. They use the same container in the first treatment arm, but without the tablets. The third one is the control without any intervention. So this is the detail of their intervention. After they conduct the study, they also use a pilot study to identify the ideal dose of the tablets, which generate the chlorine. They find that 133 milligram NADCC tablets in 10 liters of water will corresponding to an initial free chlorine dose of 2 milligram per liter. That dose was identified as adequate. We can see that they identified a commercial water storage jar with a tightly fitting lid compared to the lidless Koshi. And it also had narrow mouth with a durable tap as a suitable safe storage container. Before the study begins, they educated all the participants, the usage and how to use these containers. Yeah. Then come to the outcome definition and the measurement part. First, they conduct a study in the dry season in Bangladesh, which comes through the October of 2011 and May 2012. On average, 2 weeks after each promotion visit, they record the self-reported 2-day and 7-day prevalence of diarrhea, which defined as 3 or more loose stools within a 24 hours period in index children, or any other children's in the household which were enrolled in our study. They use negative control outcomes, which was the self-reported prevalent skin rashes and air infections to evaluate. And then comes to the question I will pose for the audience. What's the function of the negative control outcome? Actually, I have some, you can leave it in the checkbox or just speak out in our field. And I have some hints about this question because our study design is non-blinded design, and we use the self-reported outcome. So it's a hint. Excuse me? Yeah, is there any volunteers? Yeah.
To reduce potential reporting bias.
Yeah. Yeah, exactly. Okay.
So I will.
Oh, I can play sound, right? Okay. Okay. Yeah, that's right. Since we have our study is non-blinded and self-reported, it exists a reporting bias, and we use the symptom of prevalence of skin rashes and air infections. These symptoms were not affected by our intervention of chlorine, and we use this to minimize our reporting bias, and we will have the result of the negative control outcome in our result part. Yeah, and exact dry season analysis, and they also extend the rainy season visits. That's because they find that there's rain season and a dry season and rainy season in Bangladesh, and the effect of our intervention and the water quality may be different between the different seasons. So they extend the study through June of 2011 and November of 2012. During each follow-up visit, they also monitor the intervention uptake by reporting the self-reported use, conducting spot checks on the process, and they collect stored water samples in all the household, and they examine the chlorine concentration, which as for the free chlorine residues. Let's come to the statistical method part. The primary outcome they use is the 7-day period prevalence of self-reported diarrhea in index children. They calculate the disease prevalence ratio between the study arms. They use generalized linear models with log link, since the outcome was the prevalence ratio. And they use binomial error distributions because our outcome is a binomial as the primary outcome. And they use the robust standard errors to account for the clustering, because in our study, one household may have several children, and it have a clustering effect, which may make their outcome similar. So they use the robust standard errors to minimize the clustering bias. The second outcome they used was the fecal contamination of stored water. They defined it as the proportion of samples within E. coli counts exceeding the WHO threshold. They compared the stored as well as the salt water quality using the CAR-SQUARE test for the proportion of samples in these risk categories. And they also contact subgroup analysis with the season, just as I told before. Yeah, and I think that's my part of the presentation. So we do you have any question? Okay, if you have any question in the following present, you can just post it in the Dropbox or just speak out in our field. And I will introduce my partner, Nancy Huang, to continue the next part.
I will try to move it up.
So, can everybody hear me well?
Yes.
Great, thank you.
Sorry for that. Okay.
Okay, so now we're starting to talk about result part. First of all, it's the okay, that's fine. Just moving around the stuff. But the flow chart of our study participation. So if you look at the right-hand side, you can see there are a total of 1,800 of households are randomized into 3 arms. Chlorine plus safe surge arms, and the control arms. And there are 1,786 households that received promotion visits after the randomization. And throughout 1 year of follow-up, at the bottom, you can see that there are 1,649 households eventually completed the study. So as you can see, there is a total of 151 households are lost to follow-up due to either refusal, relocation, or enrolled child death. And since, first of all, like there are similar characteristics for those who leave in each arm. And also the characteristics still balance after, like for those who completed the study. So we can say that those lost to follow-ups are missing at random. And also because in each arms, they have similar refusal rate. So we can also suggest that their people have similar willingness to participate in each. And this is the table for baseline characteristics. And the total number here, as you can see, is at randomization. That's why it's 600 households in each arm. The characteristics can be based on, first of all, demographics and socioeconomics, water-related practices, and lastly, health indicators, which include our primary outcomes, diarrhea, and also our NCO, skin rash, and hair infection. Over each characteristics, we can find that 3 study arms had similar distribution. And here's the question, especially for first-year students can answer. So why is the balance across groups in RCT table 1 as important?
Well, it's exchangeability. In randomized trials, you have to respect the principle of exchangeability. So a balance between 2 groups is important for that. I'm a little, I'm sorry, I might have missed. How did you guys recruit these participants?
Oh, yeah. Let me return to the part.
Thank you.
Yeah, the investigators, they randomly select the village in this district in Bangladesh. And they use these eligible criteria for each household, and they will be chosen in the study. Does it make sense?
It does. Sorry, I had missed that. So I was going over your flow chart and I had missed that piece of information. Thank you.
It's okay, it's okay.
So now we come back to baseline. So your answer is correct. Great. So the reason of the balance is important is that we always try to limit the potential confounding, right? So by presenting that those we know, the characters we know are balanced, we can say that, okay, for those that we are not able to examine, they're also balanced. So researchers also look at the intervention uptake for the two intervention arms. It is sort of like checking the parents in medication studies. So as you can see, the both arms both achieve high uptake. So for example, when they asked the household to provide a cup of water for young children, most of them retrieve water from provided containers. And also, when we look at the delivery storage containers when the interviewer visits, most of them are filled. And lastly, when we look at the safe storage plus chlorine arms, more than half of the households have residual chlorine that above the recommended 0.2 milligram per liter. So this is our first, the result of water quality of tube well and storage water. The left-hand side is the plot on the left-hand side is the tube wells E. coli positive samples percentage, and the right-hand side is for storage water. So if we look at the left-hand plot first, we can see that each bar is they're all composed of positive samples. The difference is that when we look at the darkest proportion, that is the samples that exceed 100 CFU, which is also the limit of moderate risk. And the second dark bar is 11 to 100 CFU, which is exceeding the low risk limit of contamination. And we look back at the whole tube well water samples. Overall, there are 41% of positive, with 3% of samples exceed moderate level of risk. However, there's no difference of contamination percentage between each arms whenever we assess those who exceed low risk or exceed moderate risk. And however, in the storage water plot on the right-hand side, 89%, 17%, and 22% respectively have positive samples in control arms, safe storage arms, and chlorine plus safe storage arms. Also, if we compare each two pairs, they are all having significant differences. Now we look at the temporal trend in the storage E. coli arms only. The difference of these two plots is that the left-hand side are showing the samples that exceed the low risk limit, and the right-hand side is exceeding moderate risk limit. But from the both plot, you can see that there are similarities. So the first point is that contamination is higher in the monsoon season across arm. So the monsoon season, if we look at the follow-up round, 1 to 5 is the dry season, and after the 5th follow-up round, it will be monsoon season. And the second similar point is that the rank of contamination remain. And the highest is control, and then the safe, and then the lowest is the chlorine plus safety arm. Here's the results for our main outcome. First, we look at the temporal trend of child diarrhea. There is 1,814 total enrollment of our children. And the prevalence decreased as the study proceeded. But say it peaked at the start of monsoon, which is the fifth follow-up round. And then decreased afterwards. And so here, just to mention, the ICC for our repeated measures is 0.06. It is like the full name is intracluster correlation coefficient. And that shows that we have high variability within the child. Like within each observations. So here is the prevalence of child diarrhea. We have three arms, right? Control arms, safe storage arms, and chlorine plus safe storage arms. Here, just to say that the number here is the observation, not like household. That's why it exceed 2,000. And the safe storage arm had 1 PR prevalence ratio, which is comparing control to safe storage. And chlorine and safe storage arm, they have 2 prevalence ratios. The first one is comparing control to the intervention, and the second one is comparing safe storage and chlorine plus safe storage. So for the main analysis, there's a significant difference of comparing safe storage arms and control arms, and also chlorine plus safe storage arms and control arms. Both of them are showing a significant decrease comparing to control arms. However, there's no significant difference of prevalence between safe storage arms and chlorine plus safe storage arms. And then the researchers try to act at like interaction with seasons, which is certified as dry and wet, and also the interaction with child age at enrollment categorized in 3 groups. For the interaction for seasons and child age, you can see that if we look at the p-value here in safe storage arms and also the chlorine plus safe storage arms, we can see that there is no significance, which means that it doesn't really matter if they are in different seasons or the children are in different ages. And also, not in this table, but we also check the like the NCO's prevalence versus control arms. But as you can see, there is no significant difference of prevalence in both safe storage arms and chlorine plus safe storage arms. For example, although we can see that there is a slightly lower, like there is a 0.88 times lower of skin rash in safe storage arm compared to the control group, since the 95% in CI is across 1, so we can say that it's not significant. So here's the summary. First of all, the interventions, like both interventions, achieved high uptake. Second, the significant water quality improvements are shown in both safe storage arms and safe storage plus chlorine arms. There are 31% of significant reduction in children's diarrhea prevalence in safe storage arm compared to control arm. There is also a 36% of significant reduction in children's diarrhea prevalence in safe storage plus chlorine arm compared to control. However, there is no difference in diarrhea prevalence between the intervention arms. Although there is a slightly like 0.92 lower times in safe storage plus control arm compared to safe storage arm, but it's just not significant enough to show the difference. So one big question in this study is that why there is no additional benefit from chlorination if you're looking at safe storage arm and safe storage plus chlorine arms. So the research team tried to provide three possible reasons. First is that a safe storage alone is sufficiently reducing waterborne pathogens, which is true, which might be true, because they have proved that water quality already improved in the safe storage arm. And the second reason is that, well, the chlorine tablets, like the household doesn't have a good uptake among that. But as we can see in the uptake table, there is already acceptable free chlorine residual levels in those water samples. And also, the major participants, they do retrieve the water from provided containers, which means that there's a possibility that like eventually households just come to our places and get their water to the children. And the third reason is that the presence of chlorine-resistant organisms. Although this study are not able to test with E. coli, not able to test non-E. coli contamination. However, in the past studies, they all show that in these areas, bacteria pathogens are more often detected in the children's diarrhea stool samples. So this is not that likely to be true. So here we talk about the limitation of non-max design. As Hal just mentioned, self-report outcomes with non-max design often leads to report bias. But here the researchers try to provide two justifications. One is that the previous max trial already had similar findings with the current study. And those mass trials, because of their masking, they have like less sample sizes, which limit the power. And the second justification is that they tried to compensate the difficulties to mask by, first of all, they sent different field staff to distribute intervention in collecting health data. And second, we checked that there's no impact on NCOs. And lastly, there's no water, like the water quality is shown to be good. Now I can use the deck plot on the right-hand side to explain how the NCO and Water Quality Act. So we want to check the causal pathway between A, safe storage, and Y, children diarrhea. And we want to see that the pathway between A, U, Y doesn't exist. U is the participant expectation, which is unmeasured. So we do it by checking the A, U to W, which is our NCO pathway, does not exist, to say that, okay, so U itself is not present. We also check the pathway between A to O to Y to reinforce that the A to Y pathway does exist. So this is just how NCO act in the whole study. There's also other limitations here. So first of all, is that study findings are more generalizable to settings with good microbiological water quality and common storing water practice like Koshi and JAX. However, the research team, they do try to select places with low gram iron and arsenic concentration to maximize the chlorine effect. And the second one is, it is the efficacy trial after all. So there will be expecting higher uptake and better health impact. But this study might as well be effective enough because when they are providing intervention, especially the safe storage jugs, they try to use some easy to use aspirational products with strong promotion. And also compared to adding chlorine, they need a lower behavior change. And eventually, the objective of the study is to find out whether consistently treating and safely storing to build water would improve water quality and reduce diarrhea in rural Bangladesh. So they tried to generalize the study results to the whole rural area and lengthen the time of treating water. So this is the final conclusion. First, safe storage water with or without chlorination both significantly improve the microbiological quality in water and decrease children's diarrhea prevalence. Secondary, chlorination didn't provide additional safety, possibly due to low contamination in the water source, and also safe storage itself is effective enough. And this study also confirmed that the dominant cause of children's diarrhea is unsafe storage. The readily adopted standalone intervention was also protective against waterborne diseases in children in rural Bangladesh. And in the future, the research team is expecting to implement and evaluate a longer term of safe water storage in Bangladesh. And this concludes our presentation. You guys are welcome to ask any questions or any suggestions. Thank you so much for listening.
Thank you.
So, excellent presentation, and they were generous. I really had very little to do with this. This was all their own work. One question is, in an unmasked trial like this, I didn't quite understand the chlorine levels. Because one of the risks for an unmasked trial, of course, is that they just share chlorine tablets, and everybody in the safe storage is getting chlorine. So did they detect higher levels of chlorine in the chlorine group?
Okay, so they do see a higher chlorine level in the chlorine groups compared to other arms, which they do not throw in the tablets.
Right. Okay, so the reason I ask is just in the early trials of AZT for HIV, very early in the AIDS epidemic, patients would just, which was placebo controlled, patients were convinced that AZT was going to work, and they would actually share tablets, frustrating the ability of investigators to detect changes. Of course, the effect was so big that it didn't matter. They still saw the benefit, but it's a danger of unmasked trials that you have to be. And I think looking at the chlorine levels and looking at the ability to reduce the bacterial counts is an important way to evaluate whether or not there's a potential for confounding by non-adherence to the treatment arm.
It was unmasked, but they were also cluster randomized, no? So I guess that kind of touches on one of my questions, was how much crossover could have potentially happened. I didn't really get a sense for that in the article. I don't know if anyone else read anything about the design of the study, which unfortunately I didn't get a chance to, in terms of crossover or how they decided on the clusters. They mentioned something, but it was a very short paragraph.
So, Shariza, I'm not sure it was cluster randomized. Well, it was cluster randomized, obviously, because they evaluated all children. They randomized households, but all the children within a house were counted. So yeah, it was, I guess.
It was, yeah, it wasn't very
yeah.
clear, but it was. You have to
Yeah.
dig a little bit.
Yep. So,
I saw one thing to Dr. Jobs's point is I saw on the bottom of page 5 that in the last paragraph that they looked for samples of chlorine tested were also collected in a subset of households and of 15 in the safe storage arm. From those located closest to the households, the chlorine arm to assess for any spillovers. So I didn't see the results of that analysis, Dr. Jobs, but it does seem, perhaps it's in the supplementary materials, but that kind of addresses your well taken point about a spillover effect.
And then my other question for the methodologists is, what do you, when you have 3 arms, what do you think about randomization using a block size of 3?
Sometimes you have to with the clusters because of the way things pan out. It's unrealistic to do larger block sizes sometimes.
I think
I think it's more common with the cluster randomized trials. Sorry, go ahead, whoever was talking.
Maybe they use the block size of 3 if the study collection may be limited, or they can't have more blocks. Does that make sense? I don't know.
Yes, I agree with Hal. So like, I think in the classes, we were taught that like, if there's like 3 arms, we should choose like block size bigger than 3, or otherwise it would be limited to like, if we already randomized the first 2 and then like, it would be easily exposed that the left one is the other, or the left like intervention that we are looking at. I think this is like the issue of the block size here. Is that correct?
That's the issue, but what you've heard from Shariza is that
In practice, it's harder.
yes,
For clusters, yeah, for cluster randomized trials, it tends to be a little bit harder to adhere to that. It's also harder to detect in that scenario, just because of the way the trial is set up. So the concern is not as dire as it would be in an individual randomized trial. Your point stands though. Remember that when you have to take your comps and your exams.
Question I had was what you thought about the follow-up that they did in the control arm, and why that might have been an important part of the study design versus the fact that they followed up with the control households like once per month. Do you know if there was a reason why that happened or a certain effect they might be trying?
Actually, they have the promotion visits for the country to make sure that how they use the or how they storage the water.
I think that's a good point. So they're looking, it's kind of a promotional thing. I think one thing that the authors point out on the top of page 5 is kind of preventing this differential Hawthorne effect. So I don't know if you're familiar with the Hawthorne effect, but it's the idea that subjects might change their behavior when they're being observed. So if you are doing a study on, you know, observing people smoking during smoke breaks or something like that, they might change their behavior if they know that they're being watched. So I think that's, to your point, they're doing promotion. So by checking in on the control arms, you're making sure that it's not just the people in the storage or the chlorine arms are just changing their behavior because someone's checking in on them.
Yeah.
Yeah, oh, and just to follow on that, so that's why when in the result part, as I remember, they do mention that they when they go back and visit, they do check on like whether like where are they still remaining their like old habits of like drinking water and such.
Exactly, yeah.
The frequency of follow-up change that effect of being observed?
What would be your hypothesis?
I think it if you monitor more frequently, and they would probably be more effective.
I think you're right. I think if you were following up with the treatment arms, the sleep storage arms and the chlorine arms more frequently, let's say you only checked in on the control arm like once every few months, I definitely think that would change the outcome.
Yeah, as long as they're the same.
Exactly, yeah. Good.
Also, feel free to type the questions in the chat.
I don't see any questions in the chat looking at it.
We spent some time talking about the cluster randomized design. Just in the interest of the students being here, what are some benefits and drawbacks to that type of design? Just spitballing if anyone has any thoughts as you guys prepare for, your comps and whatnot.
Are you asking about the benefits of cluster randomization?
Correct. So why might you want to choose a cluster randomized trial? And what are some things you should be aware of?
So I can like say, so from the practicality,
Yeah,
especially in community trials, a lot of time we want to see like the effect for the whole, maybe like whole households or whole community, and that's when like cluster randomization is really helpful compared to randomizing individuals. And we also have to take account of the similarity within like a cluster, and that's also when we can do that.
absolutely. And I think Olivia's chiming in as well with the contamination between the groups. That's definitely true. I think both of you hit on the same things.
Yeah, and just to make it very pragmatic, imagine you're doing a trial of an antibiotic therapy for a highly contagious disease. And by treating selected patients, you're reducing the ability to infect other patients. So there's this huge effect. There's an effect on other participants in the trial if you want to individual randomization. But if you were randomizing, say, villages, where everybody in a village is treated, then your villages are sufficiently distinct that there's not a lot of mixing between villages, then you avoid that problem with a cluster randomized design.
Yeah. We also see cluster randomized designs used frequently in lower resource populations. So what are some ethical considerations we'd want to consider when designing the trial, when working with, if it's a drug trial, the drug companies or the intervention companies, whatever the case may be? What are some things you might want to have in those discussions? Any thoughts? Any of you worked in those types of situations or?
I was just gonna say that one thing that I noticed from the paper was that they did provide cleaning solutions after it was approved, after they saw the.
Yep, great. Good point. You hit on exactly what I was thinking about. So after the trial, you might want to consider offering the effective intervention, provided it's effective, of course, to the rest of the study population, right? Any other things regarding implementation you might want to consider in your design? How would you incorporate that? At what point would you incorporate that? especially for community-based interventions, right? You want to incorporate the input from those who are on the ground, those who are in the community, those types of things. So you'd probably want to bring them in during the planning phase for your study.
Which actually also leads to the issue of consent on these studies. Very often when you're doing cluster randomized studies within groups like this, you know, you're randomizing, in this case, households, but in other cases, villages or whatever, and you have to address the issues of consent and how that's going to be obtained.
I don't think I've catched that information, but do they consent before or after randomization?
I think they sort of mentioned about consent, so.
I remember if they did, because they said they randomly selected households. And then, is it consent after they randomly select? Because if that's the case, then whether or not the consent is balanced in randomized group would also affect.
But here I have a question, so like usually in randomized trials, do people like provide consent before or after the randomization? Like what's usually seen?
Standard is before.
Before, right?
The standard is before, but with cluster randomized trials, remember, it depends on what level you're clustering at, right? So when you're randomizing at the cluster level. If the cluster is not actually the individual, but say an organization, you're then asking the individuals whether or not they would like to participate. It's not really a consent per se, because they're still under whatever entity, but whether or not they're participating in the study is a different issue.
I've had occasion to be on DSMCs for treatments of trachoma, and they use a cluster randomized design almost all the time because of the contamination effect of the treatment. And there, very often, the consent is obtained from the village headman. And once he says yes, all the villagers automatically agree because he runs the thing for them. And so there it's consent to participate. And of course, then the villages get randomly assigned.
Yeah. It's very cultural in some contexts. But again, that emphasizes the need to actually understand the context within which you're conducting your trial, right?
Yeah, absolutely.
Any other questions or so? We have about 5 more minutes. Any last thoughts or concerns? If not, we can give you guys your 5-6 minutes back.
So my only comment is nice job, everybody.
Yep, great job. Thank you, everyone. Take care. And we're sorry we couldn't be there. We had illness and meetings and all sorts of things, but glad you guys could all join.